So, hello everyone, welcome to our presentation. And the topic of our presentation is imaging brain activity with one-photon excitation. And our group consists of Khang, which is me, and Nhat. So there are two parts to this presentation. I will introduce the introduction and background, and Nhat will introduce imaging brain activity with one-photon excitation. So first of all, let us talk about 1p excitation. So 1p excitation consists of a molecule or atom in the ground state absorbing a photon of high energy, which is represented by blue color here. So photon gets absorbed, and that photon, after it gets absorbed, will get the molecule into a higher energy, and from that higher energy, the atom or molecule will release another photon of lower energy, which is represented by the green color here, and that photon is emitted. So next up we have wide field imaging. The upper picture shows the schematics of wide field imaging, but I'm not going to go into any detail. What I want to point out though is the fact that in wide field imaging, photons get absorbed on the focal plane in a wide area, hence the name wide field. But the downside is that there can be a lot of out of focus light, which is represented in the lower picture. And so the next microscopy technique is called light sheet microscopy, and it uses a light sheet to go through a thin slice of a sample, and from that thin slice we get a 2D image. It has low photo damage and low phototoxicity because only a thin slice of the sample is illuminated at any given time. Light sheet microscopy is 2D and can be used as an ad hoc technique for 3D imaging, but it's difficult to perfectly align both light sheets in three-dimensional spaces, so you need a lot of precise calculation and modification. And last but not least, we have multimode optical fiber for imaging. So the multimode fiber has a very large core diameter compared with the single mode, but they're still very small in general, so it is good for in vivo because it's very non-invasive. So you can have the mice roaming around in the cage while you can still have pretty effective imaging data. So lastly, you can see if the multimode optical fiber in action here, and in general, multimode optical fiber imaging provides a route to achieving high-resolution optical access to deep brain subcellular processes in living and ultimately in freely behaving animals.
Hello everyone, my name is Nhat, and now I will continue for the our presentations. My presentation is about the part 2 of our presentations, and now I will talk about the wide field calcium imaging. In vivo calcium imaging, in the way my expressing suggests, allow simultaneous imaging of many brain areas as might perform the complex behavior tasks, and very much like a conventional epifluorescent microscope. A wide field or the major scale fluorescent imaging system consists of two major components: an excitation part delivering light to the brain surface, and an emission part that projects an image of the light emitted by the fluorescent indicator onto a camera chip. The advantage of wide field imaging is large field of view and the ability to image through the integral, and for the typical wide field imaging setup is fairly inexpensive, appearing to many labs worldwide. Also, it has some disadvantages like one-photon imaging is mainly sensitive to superficial cortical layer and suffer from the light scattering, and particularly when imaging through the skull. In addition, the raw fluorescent signal can be contaminated by hemoglobin absorption. The next one I want to talk about the wearable one-photon microscopy. The tabletop microscopy system, such as the figure that's shown below, requires the head fixation of the animal under the microscope objective. The wearable imaging system have enabled biological study difficult or unfeasible with a larger stationary counterpart. And in the figure here, the figure 1 is the example of one-photon microscopy for the high-speed cellular resolution imaging in freely behaving mice. The figure B is an example of the right image of the GCAM6F CAB2B indicator expressing and the computational identified cells. The figure C is the spatially tuned activity pattern across 20 example cells. This part I want to talk about the imaging in 3D light sheet microscopy. Applied to the light sheet microscopy has traditionally been applied to imaging square tissue, where generating a thin light sheet and focusing onto the light sheet with a second orthogonal objective is relatively simple for sample up to the side and a whole mouse brain. The figure below here in the part A is the scale microscopy. Use an oblique light sheet to illuminate the sample, and emitted light is collected by the same objective lens. The figure B is the experimental imaging configurations to image the neural activity in zebrafish larvae and awake. The figure C is the spontaneous activity of apical dendrite of 5-layer neuron and time course of 7 number region of interest. The last one is a deep imaging through the multimode optical fibers. This figure shows the deep brain imaging through an optical fiber. The figure here is a coherent line propagated through the multimode fiber form a spectral pattern, and other output and the manipulations of the wavefront entering the multimode optical fiber yield a desired optical field. The figure B and C is the experimental configuration for in vivo imaging. The figure D is the inhibitory neuron expression image in vivo. The figure E here is the trade-off between the fiber size and the resolution. The figure F is the dendritic beam of a hippocampal neuron ex vivo result using multimode optical fiber compared with confocal microscope. The figure G here is in vivo neuron expression of GCAF and their activity visualize a standard deviation over time. And this is the end of our presentations. Thank you for your listening.